In this video, we're going to talk about eukaryotic cells. I'm going to draw one and label it up and give a brief overview of some of the functions of the organelles within the eukaryotic cell. Now, compared to prokaryotic cells, eukaryotic cells are much newer, okay, and they make much more complex organisms, okay, like people or plants or fungi, okay. Now, the key difference between a eukaryotic cell and a prokaryotic cell is that it has its genetic information within the nucleus. So they have a nucleus which holds the genetic information. And they have lots of organelles that can be membrane bound. So they have membrane bound organelles. Okay. Not all the organelles are membrane bound, but a lot of them are. And that's the difference between eukaryotic cells and prokaryotic cells. The big main differences. Eukaryotic cells are bigger and, like I say, cause more complex, make more complex life. But these are the key things. All right. So I'm going to start off by drawing the cell, and I'm going to draw some images. Now, they're not the most accurate images that I'll draw. I don't profess to be that amazing at drawing. However, I'll give explanations on how to identify them in different images. Okay, and at the end, I'll show you an image of a cell, so you can kind of put what I've said into perspective. So, what we have is we have our cell. I'll be a bit lazy and do it that way. Okay, and here we have our cell, and it has its cell membrane. Okay, which is this outer layer here, and that holds everything in. I've drawn a round cell here, I should say. This is going to represent an animal cell. We've got plant cells and all different kinds of cells, but this is just a general overview of eukaryotic cells. So we have the cell membrane, which is more than just a border. It's semi-permeable, which lets some things in and some things out, what the cells need. Okay, but that's a whole different topic. And then what we have is like a jelly-like substance in here. In some textbooks, they'll call it the cytosol, which is what the name of the jelly is. But the combination of the cytosol, the jelly, and all the things that are inside the cell, all the organelles, are called the cytoplasm. Okay, so this grey substance is the cytoplasm, which includes the jelly and the organelles. It can be confusing depending what text you're reading. As I said before, one of the first important things is the nucleus. Is the nucleus. And the nucleus isn't, sometimes you just see it in things where it's just a little dot. It's not just a little dot. So the line here I've drawn is called the nuclear envelope. Okay, and the holes, the gaps within the nuclear envelope here, these gaps, called the pores. And what happens, there'll be genetic information stored in the nucleus, but that information needs to leave, and it leaves the nuclear envelope via these nuclear pores. You'll also see a dark spot, which is called the nucleolus. Okay. And this stores a certain kind of RNA called ribosomal RNA, which is the code on how to make ribosomes. Now, depending which textbook you read, again, you'll see either dots within the nucleus, which is known as the chromatin, or you'll see actual chromosomes. Okay. You won't see both together. Okay, but they're both the same thing. They're the genetic material within the nucleus, which codes for all the proteins that make an organism the organism. Okay, so maybe a little bit more complicated than you've seen before. Just get rid of all this. What I'm going to do is stick our nucleus, squish it down a bit, stick it in the cell. Okay. The next organelle we'll talk about is a set of organelles called the endoplasmic reticulum. Okay, and you get two types of these. You get the smooth endoplasmic reticulum, which is written as the SER, and you get the rough endoplasmic reticulum, which is written as the RER. Okay, they're both the same thing. Some books or images it'll just show this. Okay, both are membranes that are filled with fluids, or set of membranes that are filled with fluids. So we'll draw the smooth one first. And they both synthesize certain things. So they're basically just, you'll see an image like this of just some stuff laid upon each other. And they're just sacks of fluid. Okay? They're not just sacks of fluid, they do more things than that, but from our point of view, the sacks of fluid. And the smooth ER processes lipids. Okay, that's its job. So the rough ER is almost the same. Okay, it still has these membrane-bound sacks of fluid, but what they also have are dots on them. And it's these little dots that make it rough. Okay, it's these little dots that make it rough, and these dots are ribosomes, which synthesize proteins. So the smooth ER synthesizes lipids, and the rough ER synthesizes proteins. So put our smooth ER here. Okay, I'll stick another one in. There's not just one set of these organelles. And the rough ER, when you'll see it on an image, it's always stuck onto the nucleus. And the reason it's stuck onto the nucleus is because the information to make proteins comes out of the nucleus. It doesn't want to wander around the cell, so it goes straight to these things where the ribosomes that are on the rough ER, which can then synthesize these proteins. So you've got the smooth ER and the rough ER. Do them in brackets. Okay, two of them. The next organelle I've already briefly spoke about, and I'm just going to draw it in here, is these red dots. Now these red dots will be the smallest thing you'll see in an image, and these are the ribosomes. They are made up of two units, made up of two parts, and basically these clamp on to information that comes out of the nucleus, and then they make the protein. They build the amino acids that make the protein. You get fixed ribosomes, which you'll find on the rough ER, and you get loose or freely moving ribosomes, which are just in the cytoplasm. Okay, so they make proteins. The next thing we're going to talk about is what's called the Golgi Body, or the Golgi apparatus. And the Golgi Body or the Golgi apparatus, depending again what text you're reading it in, are similar construct to the smooth ER as the fact that they're membranes that have fluid in them. So, and I've seen images that make them look very similar to the smoothie are, very confusing. But what the Golgi does, lipids that are made from the smoothie are and proteins that are made from the roughy are packaged in here and then moved around the cell. So these packages then travel through things called the vesicles, and vesicles around things. So the Golgi, when you see an image, will have these round things around it. Okay, and these are like the post leaving the Golgi. So this would maybe have a protein in it or a lipid. So the Golgi, you'll see these round things around them. But you can see how people get confused with this and this. So that's the Golgi. As I said before, you've got these circles, these bigger circles in the ribosomes, and they're called the vesicles, and they carry things around the cell. Okay, so they're there. They come from the Golgi, they'll carry your proteins and stuff like that. The next thing I want to talk about is what's called the lysosome. Or lysosomes, and these are even bigger circles than the vesicles or the ribosomes, and they're normally clear. So the biggest circles you'll see where there's lots of them are called the lysosomes, and what they do is they carry digestive enzymes which break down products, sometimes foreign products, but they can recycle organelles within the cell that need to be remade or are too old or something like that. So lysosomes carry enzymes within them that can break down things. The other thing we've got is what's very special towards eukaryotic cells is the mitochondria. The mitochondria. Okay. Now mitochondria is a double, in fact I'll draw this a different colour so you can see it a bit better when I stick it in there. It has a double membrane, so not just a single membrane like the smoothie or the roughy or the Golgi, has a double membrane, and it has kind of like these grooves inside them. Now, if you know anything about respiration, cellular respiration, you'll understand what these are, okay, but I'm not going to go into them. I've got another video that talks about cellular respiration, well, a set of videos. But basically, what the mitochondria does, when an organism has its glucose, it can't use the glucose as energy within the cell. So the mitochondria transforms that glucose energy into ATP, adenosine triphosphate. Okay, and then that can be used within the cell. Depending on the kind of cell, will depend on how much mitochondria you get. Well, a muscle cell would have lots of mitochondria because they use lots of energy, for example. Okay. So this would be in an animal cell. The other standard organelle we see within eukaryotic cells as a whole, okay, but not in an animal cell, you would see in a plant cell, is the chloroplast. And the chloroplast isn't much different as in the structure to the mitochondria. Again, it has a double membrane, but this time it has stacks of circular things on. And again, in my photosynthesis videos, the structure of these is explained more, but I'm just drawing this like this to help you identify it when you see it on an image. And a chloroplast job is animals eat glucose in the form of food, and then that glucose gets transformed using the mitochondria. Plants don't eat food, they create glucose from the sun, and they use the system photosynthesis. Okay, you can see some of these in bacteria actually, but they're not as well formed. Okay, so the chloroplast is what you'll see in algae and plants, and they what make that cell's glucose, and then that glucose, like the food glucose would go over to the mitochondria. Okay, so we've got our nucleus, our endoplasmic reticulums, the smooth and the rough, ribosomes, the Golgi apparatus, vesicles, lysosomes, mitochondria, chloroplasts. These are your bog standard organelles that you find within eukaryotic cells. Obviously, chloroplasts aren't found in animal cells, and depending on the cell's function, you'll have different numbers of these organelles within them. Okay, but this is just an overview. There's also more organelles you could see. Now, this is an image I got from Google Images, and it just shows you an animal cell, and hopefully you can label the things. You can relate the image here to the drawings. So we've got our nucleus, which has its nuclear envelope and its pores. We have the nucleolus, which is this darker spot. We have this rough looking stuff attached to the nucleus here, and that's the rough endoplasmic reticulum. And here you have the smooth one, because it doesn't have the dots on. These dots that are on the rough endoplasmic reticulum are the ribosomes, and you can see that they're also freely moving within the cell. You have the Golgi here, and it has lysosomes leaving it, these circular things leaving the Golgi body. Okay, the lysosomes. Okay, and it's identified there. You have the mitochondria, which you can see has got the folds in, as I said. And the other thing that you've got in here that I didn't talk about in the other one is the cytoskeleton. Now, the cytoskeleton basically just gives the cell its support. If you just have the cytoplasm in it, it'd just be all floppy like a water balloon full of water. But the cytoskeleton acts like the cell's scaffolding. But I haven't spoke about it in the video before, in the images before, because it's not in the A-level specifications. Okay, but if you see the cytoskeleton, that's what it is, and it doesn't really have a place here. Okay, as I said, this is just some organelles. There's more organelles, but this is just some.